This is the third part of the lecture for week 1 of biology in the second quarter, talking about the chemistry of life. And we're going to finish up talking about the most common and most important biological molecules in biochemistry. We're going to finish that up. We're going to finish up at this point the discussion on carbohydrates by talking about the reactions that can occur with carbohydrates. And there were two reactions that were discussed in your book. One is called a dehydration reaction. A dehydration reaction is a chemical reaction that occurs in which two molecules come together and remove a water molecule and end up being linked together. And the diagram that you're looking at is a example of a dehydration reaction. And what you have is the combination of a substance that generally has a OH or a hydroxide group on the side, like this one here, and it combines with this hydrogen here and forms water. And at that time, when these two are removed, then this carbon links to this nitrogen in this case to form this larger molecule. And this is an example of a dehydration reaction. Now, if you can imagine, if the arrow is turned around this way, pointing in this direction, and the water comes in and breaks apart this to form these two compounds, then that's what's known as hydrolysis. Hydrolysis is the breaking apart of a larger molecule by adding a water molecule to that molecule. Now, here we have a substance called carboxylic acid. And an acid is any compound that when it is dissolved in water, it will release a hydrogen. And this hydrogen is released as an ion in the solution when this is dissolved in water. So this will react or will combine physically with the water, and this is released to form an ion of hydrogen and then an ion of the rest. If, on the other hand, say this whole hydroxide were released, which is not what happens, but let's say it was, then this would be known as a base. A base is a compound that releases hydroxide ions in solution. And that doesn't occur in this example we have on the slide here, but if it were to occur, then it would be called a base instead of an acid. Now, let's move on from carbohydrates to lipids. What is a lipid? A lipid is a combination of glycerol and fatty acids. Okay, here is an example of a lipid. It's called a triglyceride because it's 3 fatty acids, thus the term tri, attached to one glycerol. Now, glycerol by itself just has a hydrogen atom here, a hydrogen atom here, and a hydrogen atom here. But when it combines with the fatty acid, that hydrogen is removed. Okay? If you can remember back on this last slide, you can take a water molecule out and combine two together. Okay? So now if you take the hydrogen atom from this and an OH from this acid, fatty acid here, and link them together doing a dehydration reaction, then you end up with a lipid. So you do it 1, 2, 3 times. Now, fats actually come in 2 forms. Lipids come in 2 forms. You notice that at the top here, all of these have single bonds and as many hydrogens as it can possibly have. That is known as a saturated fat. A saturated fat is if all 3 of these were to have single bonds all the way across. But you see here there's a double bond. Because there's a double bond, that's called an unsaturated fat. So in fats that where all 3 of these have all single bonds That's known as a saturated fat. What it essentially means is it's saturated with hydrogen. But because it has this double bond, it's called an unsaturated fat. Now the term I skipped here is called hydrophobic, and you remember we were talking about this back when we were talking about plasma membranes, that this part of this molecule was called hydrophobic, meaning it had no affinity. It will not mix with water. And we all well know that lipids or fats or oils do not mix with water and therefore are known as hydrophobic. Now the next biological chemical is called a protein. Proteins are made up of basic building blocks called amino acids. Amino acids have a nitrogen containing group, which is the amino part, and an acid group, which is the acid part. And these are linked together in long strings to form what is known as a protein. The bond or the linkage between amino acids is called a peptide bond. And every protein has three structures. The first structure is the one-dimensional, just the sequence of amino acids. There are 20 different amino acids, and each protein has a unique sequence of what amino acid is linked to which amino acid. And that linear sequence is called the primary structure. Because of certain chemical interactions, this will then begin to coil up in a two-dimensional structure. Usually it's either a alpha helix or a beta pleated sheet, and that is known as the secondary structure. And then finally it will form a glob, which is known as the tertiary structure. This is a three-dimensional structure, and this is actually extremely important in the function of most proteins, is its actual three-dimensional configuration, which is why in some situations, if you change just one amino acid in this sequence, this changes and this changes, and because this three-dimensional structure changes, the protein does not function properly. And here is why. Specialized proteins that act as catalysts for biochemical reactions are called enzymes. And this is an example of an enzyme. And what you see is this three-dimensional structure forms a little pocket where the substrate, which is the part of which is the chemical in which the enzyme acts on, can fit into this and form a complex. Now this whole process, this pocket is called the active site. It's the area of the enzyme that's responsible for where the chemical reaction occurs and where the speeding of the reaction occurs. So this active site, the substrate comes in and binds in this active site and then forms this complex. This is known as the lock and key theory of how enzymes work. And it's a structural theory. So if you imagine that if this structure were to change in some way, and this pocket were to change in its shape, then the substrate wouldn't fit in. It would be like shoving peanut butter or bubble gum into a key lock. The key can't go in anymore. And so this is a more stylized version of the same thing, showing that the conformation or shape of this pocket or active site is critical in allowing this piece to fit in to this situation. If this shape changes, then this piece won't fit and the enzyme no longer works. And that is why many times if the enzyme is changed in some way, there's a devastating result from that. The last biological molecule we're going to talk about is called DNA. And this is just how chemically DNA works. DNA is made up of three pieces. The foundational building block of DNA, is made of three pieces. And that is called a nucleotide. Now, actually, nucleotides make up both DNA and RNA, but we're going to specifically talk about the DNA nucleotides. And the DNA and the nucleotide is made up of three pieces. The first piece is the deoxyribose sugar, which is right here in the middle. And this is the sugar part, and it acts as the middle part of the atom molecule. Also attached are these nucleotide bases, also known as nitrogenous bases, obviously for all the nitrogens that are on there. And there are actually four of these: adenine, guanine, cytosine, and thymine. Thymine in DNA and uracil in RNA. And the bases are extremely important because the bases are where the information is transferred, the sequence of the bases. And we will talk about that when we talk about how genetic information is transferred. The third part is the phosphate part. The phosphate is the backbone of the molecule, and these are attached one to another in a long string and form the molecule DNA. And here's an example of how this is done. And the DNA, here's one strand of DNA, here's the other strand of DNA, and they link together. This is the molecule of DNA here, and these strands swirl around each other like a spiral staircase. And the steps of the staircase are these linkages between the bases, and the actual connection of the staircase is the bonding between phosphate groups. And you'll notice that opposite every C is a G, and opposite every A is a T. Each of these represents cytosine, adenine, thymine, and guanine. And it goes down. Here's another C with another G, and another A with another T, and this time a T with an A. And that sequence is preserved, and this is how the genetic information is transferred, is the sequence of these various bases. And we'll talk about that, as I said, when we talk about how genetic information is transferred. But the bases are bonded to each other specifically, and you'll see that there are 3 hydrogen bonds between a G and a C, and 2 hydrogen bonds between a T and an A. A hydrogen bond is a strong attraction between hydrogen atoms and certain other atoms in specific molecules. And so because there's 3 here and 2 here, a T can never combine with a C, and an A can never combine with a G because they don't have the right number of hydrogen bonds. And then this sort of sequence goes on for long strands. And this is called the double helix because it is 2 strands wrapped around in a helical structure. As I said before, the thing that forms the backbone here is the phosphate groups.